So let's take a look at translation. Now that we have the information copied into a transcript, we send it out into the cytoplasm, and now we want to make a protein. And so that's what translation is all about. To start with, we need to consider a few things. First of all, translation is the process of interpreting the information in nucleic acid language and translating it into the language of proteins. That means amino acids. Okay. When we talk about the term the genetic code is like our translation dictionary. It's the correlation between the languages of RNA and of protein. And since proteins are made of amino acids, and as we'll see later on, the sequence of amino acids, the specific amino acids involved are important, then that means that the information in messenger RNA must code for specific amino acids. And we know that about codons. Codons are those three-letter words in the transcript, and each codon must code for a specific amino acid. So when this was all sort of being discovered, this is what it looks like now. Okay, you've got the DNA strand in blue on the sort of in the middle of the page, the transcript copied directly right next to it in red, and then next to that are the codons and the specific amino acids that go with them. Now, of course, we haven't taken into account introns and exons here in alternative splicing or any of that, but that's basically how it works. Each codon in the messenger RNA corresponds to a specific amino acid, and so the sequence of bases in messenger RNA determines the sequence of amino acids in your protein. And your protein's properties, everything that protein does, comes from, at its base, which amino acids are there and what order they're in. So some things that we have to kind of figure out as we were developing our knowledge of this, and some questions that were being asked. The first question is, how many RNA bases in a transcript specify one amino acid? We know it's 3 now, but we didn't. When they first were trying to figure this out, they didn't know it was 3 bases in a codon. Is the genetic code overlapping? We'll take a look at what that means. Can messenger RNA codons signal for anything other than amino acids? Do all species use the same genetic code? And finally, which codons are the ones that specify which amino acids? So number 5 is the development of the dictionary, the genetic code. So let's take the first question. How many RNA bases specify one amino acid? Well, there are 4 bases, A, G, C, and U. There are 20 amino acids used to make all proteins. So all proteins in all life on this planet are synthesized from the same 20 amino acids. Well, clearly, we can't use 1 base per amino acid because there's only 4 bases and we need 20 amino acids. So that's not going to work. Somebody suggested, well, why don't we just put like 5 amino acids coded by A, 5 coded by C, 5 coded by G, and 5 coded by U? Well, that's going to be really confusing, because if you see an A in your transcript, how do you know which of the 5 amino acids to bring in? There's no thought process here. It's all chemical, so it has to be something that's going to happen automatically. So this isn't going to work. Well, what if we use 2 bases per codon instead? Well, the different possible combinations of 2 letters out of those 4 is 16. There they are. Those are the 16 possible ways of combining. Even if you reverse them, you still only have 16. And then we need 20 different possible letter combinations, so that's not going to work. When we go to 3, you get 64 different possible combinations. That's way more than 20, but that's good. It's better to have more combinations to cover a fewer number of amino acids, because what that means is that it's very likely that we're going to have some amino acids that have multiple 3-letter combinations. Now you might think, well, why is that any better than having multiple amino acids per letter? Well, think about it. If you have multiple ways of saying the same amino acid in your transcript, you have much, much more protection against mutation. If something happens to your transcript and a letter is switched, and it's the wrong letter, you might be okay, because that new 3-letter combination might still code for the same amino acid. We call that a degenerate code. Multiple 3-letter codons coding for the same amino acid. That means it's a degenerate code. Okay? Now, logically, this makes sense. Mathematically, it makes sense. But is that really what's going on? So we have to do some experiments. And you remember Watson and Crick, right? Well, Francis Crick, in his lab associates wanted to test to see if 3 bases will produce 1 amino acid, will code for 1 amino acid. So in order to do that, you need to know a sequence of bases that you can test. And so they used a virus gene The virus is called a T4. It's a bacteriophage. It attacks bacteria. And one of the genes in that bacteriophage, that T4 virus, was well studied. They knew the sequence of it. It wasn't too long either. So that they were going to then take that and produce a protein from it. What they did then was they produced the protein that was coded for by that, and then they took out one base. They excised or cut out one of the bases in that gene. And when it did that, it disrupted what's called the reading frame. I'll show you what that looks like in just a second. Okay? If they took out two bases, they still disrupted the reading frame. In other words, they weren't getting the right protein. It changed the entire sequence of amino acids. But when they took out three bases, 1, 2, 3, right in a row, the protein was almost fine, except it was missing an amino acid, or if they added three bases, it had an extra amino acid in there. So that told them that it was likely that the messenger RNA transcript was being read in three-letter words at a time. Here's what it looks like, okay? So here's a sentence. The fat cat ate the fat rat, okay? Our reading frame is one three-letter word at a time. Each word in this sentence is 3 letters long. I've color-coded the letters in fat because we're going to remove them one at a time. I'm going to take out the F first. I'm going to remove that base, that letter. And when I do that, we start off, okay, the fat cat ate the, and then things get weird. Then it's hard to understand. I don't know what the fat cat ate. I don't know what atra and at are. I can't read the sentence anymore. I removed one word. If I remove two letters, I take away the F and the A, well, now I still can't really understand the sentence. But when I remove all three, now the sentence is readable again. That's what I mean by reading frame. And that's what Francis Crick and his cohorts did. They removed one base from this gene, and this protein was messed up. They removed two bases, the protein was messed up. And they removed three bases, the protein was fine, but maybe it was missing an amino acid in that spot where they removed it. Okay? So the second question is, is the genetic code overlapping? The question means in a sequence of bases on a messenger RNA, is every 3 bases a codon, or do the codons overlap? So what do we mean by that? So look at this sequence, AUC, AGU, CUA. Is that 3 codons, AUC, then AGU, then CUA? Or is it maybe 7 codons, AUC, then you take the U and the C and you use them again in the next codon, A, and then the CA and the next letter, G, then the AG and the next letter, U, and so on and so forth. Now, it makes sense that if there were 7, you could pack a tremendous amount of information into just a few bases. Okay? Those 9 letters could code for a lot more information than just 3 amino acids. The problem is you limit yourself, because if you do it that way, okay, if that's the sequence, AUC, AGU, CUA, then the first amino acid, which is coded AUC, has to be followed by an amino acid whose first 2 letters are U and C. That limits the number of amino acids that you could possibly put into this protein. So it's too limiting. So that means there must not be any overlap. Every 3 letters individually create 1 amino acid, or code for 1 amino acid. Next question, can messenger RNA codons signal anything other than amino acids? Yes. Punctuation. There are directions for starting and stopping. Whenever you see AUG, that means start. It also actually codes for methionine, which is one of the amino acids sometimes. But UGA, UAA, and UAG are stop codons, like periods in the sentence. So it's kind of like punctuating the process. Do all species use the same genetic code? The answer to this is yes. All life on this planet uses the same 4 RNA bases, the same 20 amino acids. All of our proteins, now the proteins are maybe a little bit different for each species because it has to do different things, but there are some proteins that are shared among all species. There's a protein called ubiquitin, and just with the name ubiquitin, it means it's everywhere. And it has the same amino acids in there. So the importance of this question is that if we all have the same amino acids and the same 4 bases in RNA and the same sequences code for the same amino acids, that means we must have had a common ancestor at some point. The genetic code, the sequence of bases in each codon that codes for a specific amino acid, is universal on this planet anyway. So messenger RNA we can take out of one species, we can put it into another species cell, and it'll make the same protein it would make in the original species. This is what recombinant DNA is. Recombinant DNA is where we take messenger RNA from one organism, we put it into another organism, and we get it to express. We can also do that with DNA. You did that with the bioluminescent bacteria. You made them glow. You took a gene from one organism and you made the protein in another organism. Proof right there. You've done it experimentally. Which codons specify which amino acids? This is the trickiest question of all, okay? Because this is going to require a lot of experiments. Marshall Nirenberg, who at the time was in charge of the National Institutes of Health and ultimately won a Nobel Prize for this question, he and his co-workers devised a series of experiments using E. coli bacteria's machinery, the enzymes that are all responsible for this. And they made transcripts with specific sequences, and then they watched to see what kind of protein would come out. And doing this over and over again with all different variations, they were able to figure out the entire genetic code. They were able to figure out which amino acids were coded for by which codons. So here's how they did it, basically. So they started with a synthetic messenger RNA strand, all uracils, all the way across. U. So every codon is 3 U's, U. When they made the protein from that, they got phenylalanine all the way down, a long chain of phenylalanine. So right there, you know that U means phenylalanine. They did it with a strand of A's. They got all lysine amino acids, so A must be lysine. They did all G's, they got glycines. And they got all C's, they got proline. So now they know 4 of the 20 of the 64 possible combinations. And they just kept doing that. They kept making new messenger RNA strands with repeating 3-letter units and looking to see what proteins, what amino acids would come out. And what they ended up with was the genetic code, a way of predicting, based on a sequence of 3 bases, which amino acid is going to come out. And we know it's a degenerate code because some amino acids have multiple 3-letter combinations to produce their protein. Here's what it looks like. This is the chart for the genetic code. The way you read it is on the left-hand column, you read the first letter of your triplet. Across the top is your second letter of your triplet, and down the right-hand column is the third letter of your triplet. So if I pick 3 letters at random, CGA, start in the left-hand column with C, go over to G and all the way to the right, and A, and that's arginine. Each amino acid has 3-letter designation to simplify writing them. So arginine is simplified or abbreviated ARG. I can use this to find the sequence of amino acid. You'll see the stop codons in there, UAA, UAG, and UGA. You'll also see, if you look down here, AUG is methionine, MET, but it's also start. AUG is also start. Okay? Here's another version of it, and you read from the inside out. So CCA would be C, then A, proline. CC anything is proline. See, there you go. There's an example of the degenerate code. If you have CCA and somehow the A gets mutated to a U, it's okay. You still got the right amino acid in there. So it protects you from the effects of mutations. Okay? A lot of information in this video. Make sure you watch it, pause, rewind, get some good notes, do your summary, and practice decoding some 3-letter triplets with these charts. See which one you like better.